Good morning, everyone. We'll be talking about and discussing about the sample processing, which is a critical part of the MRD, basically, to have a very good data and interpretable data, the clean error-free data. So the topic is tips and tricks for getting appropriate data, but we'll basically getting on the troubleshootings of the sample processing. So how to achieve the appropriate data, because you should have the proper those series on that SACC large difference in the mean channel values for scatter side scatter always, because they are the parameters which gets affected on the sample processing. Then minimal cell loss and the clumsy cell suspension always get issues with the high where we acquired 5 million cells. Preservations of chlorochrome brightness then depend on the how the titer has been done, how the cocktail validation has been done. Low background staining along with your maybe using the many brilliant violet dyes, minimal instrument or inter-laboratory variations between the two instruments, easy and fast performance due to there, error-free acquisition and clean data plot minimum, and having the minimum non-specific binding. So what are the common issues? The basically common issues are presence of dead cells or the maximum aggregates, debris, partial red blood cells, lysis issues are there, like the resistance RBCs are there in the post-induction, post-consolidation, marrow when processing about the sample processing. High background and non-specific staining is also there. Cell yield, the issue of cell yield, you should have the proper cell yield out of the water you are rising the volumes. Change in scatter properties like forward scatter and side scatter, because of there are so many issues because of the factors, and the weak fluorescence intensity or no signals. So basically, we're talking about the first factor is reagent preparation and validation. So always keep in mind either you are using the lab like in-house preparation or commercial. So you should always have the reagent validation, so verification. So every day you get your reagent and just run. So look for the background whether it's working fine on the any normal sample, and then you go for the your proper samples with the MRD or what you are doing the performing the test on. So there you are might be using. So how many of you are using the bulk light wash protocol, or anybody is using stain light wash?
Use bulk light.
Majority of them using the bulk light. Okay. So major reagents are like lysine reagents and the PBS, and you might using the paraformaldehyd. So we need to be checks if you are in a house that it has to be validated first, and then you can process on the patient sample. So always do it on the normal samples, maybe the paraformaldehyd. Obviously, antibodies, cocktail titration, validation is very important to have the proper data. So this is just an example. If you have the lights with the 7 minutes, 2 minutes, and the sample to the rise ratio. So we have done the experiment for the incubation. So you can see here, if you have the sample to the rise ratio is very less, like worldwide that 1 part of the sample has to be diluted with the 9 part of blood. So if you have entries, as Euroflow had said that always you can go up to the 25 times lysing. So that is the better to lyse the sample. Even the time is also important. So everything is optimum. So you have to have your own experience in your lab, and you have to validate or verify the timings and your centrifugation feed, your time for the speed, and even the as per the sample also. If you have the more sample, you more go to the big tubes like 15 ml, you can lyse the sample, you can go to the 50 ml as per the Euroflow protocol is available. So you can see here, you can see the 7 minutes incubation shows the very good lysing, where you can say 2 minute incubation is not all the cells are living, so interference of RBCs are there. This is also the sample ratio, where we can see in the first plot, we can see the light ratio is not maintained, and in the second plot, light ratio is maintained. That's why we have the less number of RBCs interference. Then again, the PBS contamination always happens in-house, or you have the commercial PBS also. Anybody is using the C, so it gets exposed to the O2 oxygen, so it oxidizes and pH going to change. So that alters your results while the sample processing. This is just an example. So we just ran. You can right hand side. We can see here only we are on the PBS, because it might happen the distal water is also interfering the PBS. So this is one more issues again. We have checked for the instrument, reagent issues, sample, aging, everything. And one day we have that for a particular day we got to know that you can see on the FAC issue also there is a shift on the FAC because the window is created for the normal validation purpose, and you can see the window that forward scatter has been compromised on that. So upon knowing that was the old PBS and the new PBS, this is the way you have to validate each and every day. So we do validate our all the reagents each and every day for the purpose. Before we go for the patient sample, you always do it on the normal sample. So that's how you can do the old PBS versus and the paraformaldehyde also for the. I mean, you can see here on the left, the paraformaldehyde is corrupted, like that is affected something, it's got contaminated. That's why the forward scatter has been compromised. You can see the proper separation on the right hand side. Pardon? Old on the previous day was already made in the, because working is always made fresh. Okay. So in our scenario, it's an in-house previous. We do home brewed. Pardon? Yeah, the stock has been preferred, 5 stocks have been preferred that will last for the week. And every day we used to have the working solution. But though we have like yesterday is the stock solution, yesterday somebody has to make the, so before starting with it, we'll have to validate it. So that's why the importance of reagent validation. There are many factors which are going to be, you can see on the left hand side, on the forward scatter, the lymphocyte, granulocyte on the, they are adjacent to that. But on the right hand side, the lymphocyte is a proper discrimination between the forward scatter, the blue on the lymphocyte, on the time plot only you can make it out. So that's how the importance of the reagent validation always before going for the patient sample. This is the same thing. I'm not going into detail because you should have your own template, whether you have the predefined for the lymphosome and grants, you will come to know whether your validation of the reagent is done properly or not. So again, that is for the validation. So we have in our any flow cytometer or anywhere we can acquire on anything, and you have the predefined template to know whether this is properly working or not before going for the patient. Second thing is coming from the cell count, because cell count is important to know how much sample has to be live, always or otherwise for the stain laser method is very important. As we are acquiring 5 million of viable cells for all the MRDs except AML MRD, because it's being only 2 million cells. Otherwise, you should have only the point has to be taken, like message has to be clear here that for all the surface staining, you actually there is a loss of the cells up to 50%. But if you are TMRD, like cytoplasmic staining is there, for myeloma MRD, you should have increased the sample volume to get the appropriate or your achieved required number of events. Maybe up to the 5 million. So this is the procedure. What we do, we take the sample, we dilute with all the previous or any, you can dilute with the sheet also, or any diluent sample you have in your machine, in your lab. So 40 microdrop sample and 160 microdrop diluent can be taken and just acquire it on your analyzer, and you get the count. You require 5 million cells, you put the 5 million cells divided what you get the per microton, you will get the volume of samples. Like in the given sample, you can have the 382 microton sample, but 382, so you can double the volume to considering the even loss of, because every wash you will some loss of the cells, even viability, because of the viability, you will going to lose the cells. So we have made some Excel sheet to avoid the errors, because sometimes the operator or sometimes technologists can make a mistakes while calculating the sample volume. So you should have the predefined template. Okay, you just put the sample, it will calculate for how much. We have so many panels, like we have CLPD, we have acute leukemia, we have MRD for the lysine volume as per your assay. The another method is to have, you can also have, instead of using 20 microdrop cells, you can use 2 microdrop cells. You can take out any 2 microdrop cell, you sample ratio. You just put some lysine up to what the ratio, like 2 microdrop, you add 50 microdrop lysine, 20 microdrop lysine. Keep it for 7 minutes and just acquire it on the flow cytometer. So you will get again count. So what we did here, the small experiment that you can see here, the left hand side, the count of this 3-part counter, and this is counter on the actual count of the flow cytometer. So what we get the count shown is by like this is 9.5, this is the 19,500 because that is the 5-time dilution basically. This is the 16,000, it's like that. So what you get on the, you can see on the first plot on the 3-part pointer, there is the RBC, NRBC interference. On second also there is NRBC, and only third sample has very less amount of NRBC. So due to that also your calculation can go wrong and end up with the less number of cells. So that's why you can have the 2 microliter sign instead of 40 microliter, you can use the 2 microliter. Whatever you are getting the viable cells divided by 2, that is your true count. So that can be done. So this becomes also your answer. You can add a CD45 to more specific. Third tube is what types of you are using the tube, like you are using the Falcon tubes, are you using the like there are polystyrene tubes, you can use the Falcon tubes. There are many companies which provide the tubes. So we have did some experiment and we seen that there is a little bit difference of getting sample loss when you using the different types of cells. So it can be sometimes static adherence. So some of the cells tubes, because quality of the tubes plastic is very bad, so that can adhere the some of the cells. The DNA will go expose it and that will clear the clump also. So this is just a number. We did some experiment to know what was the expected count. So if you light the 400 microdrop samples for these 3 different tubes, so this is how the count is going to get. So even you can see the BC has a better number in this 400 micro. If you light the 550 microdrop sample, the expected number is 4 like and what to get in the 3.1, 2.8. So there is variation a lot, but this has to be carried out in your lab. So before you can always refer to the any data, but you can take this. So even you can try in the 15 ml tube to get the live sample. Even if you can try in the 50 ml tube, 15 and 52. But as per our experience, we have seen that 50 ml tube has a more loss compared to the 15 ml tube. So third, one more factor which affects your loss, but not, I don't know. So everything is optimum. So you should have optimum flow rate while acquiring the data. So if you want to acquire the 5 million cells and at the rate of, this is up to rate of, I think
113. 113.
Yeah, event rate is almost 5,000 per second. Okay. So if you want to, so it will take 15 minutes. So your technology is smart enough. If you increase the rate, so your abort rate will be the more. So again, that with the loss, you will gain going to the loss of the cells with because of the abort rate. So that is. So what is the abort rate? If flow cytometer cannot identify, I cannot generate the voltage plus, that will abort the cells. So doublets you can show that if the 2 cells are going, that will be maybe considered as a doublet. So more clump or more simultaneously 3 more. If you increase the rate, the sample core will be more. So simultaneously 3-4 cells, so it will abort it. So this is why the flow rate is 5,000 and abort rate is minimum, that is up to the 4%. So if the flow rate is 8,000, you can see the abort rate is increased also 6.5. Even if you can see on the forward scatter, side scatter, and the rest of the thing data, there is something messy. If you go still high at the flow rate of almost 11,000, so abort rate is 11%, 11.7%. You can see that on the event per second, and abort rate is given there. So that happens in each and every flow cytometer. So abort rate is the electronic abort. It's called electronic abort because that particular number of cells cannot be measured at all, taken into consideration. That will go in the waste. So this is the one way you are losing your cells number. So apart from this, there might be with a clinical history, the patient can be with the viral loads, a cold and wall antibodies. So you should treat the sample at the 37 degree. That can be a issue like the cold agglutinates. What anticoagulant, whether you are using the proper tubes, that your EDTA tubes are getting expired, that you have to look for because of that also it's getting clumping of the cells because each and every sample maximum you are getting, everybody is getting an EDTA, right? Not in the expiring. Then again, you have to work on the stability. Then even transportation also plays an important role to get a proper sample processing. While sample processing, you get the proper cell population, and again you are end up with the higher number of events for the MRD. And how do you storage your sample? That is also important. This is one of the example where I received the sample after 72 hours of the collection from the outside. And you can see on the left hand side, the first scatter, there is discrimination between the lymphocyte and granulocyte. Everything is got messy, and everything is got done. And even viability, you can say 58% viability. The same sample you can see on the left hand side, and we got this repeat sample within the 24 hours, and you can see the how is the fault scatter and side scatter. You can see on the left hand side for time versus fault scatter. So there is a proper discrimination between the lymphos, monos, and granules. And even viabilities in this is also very good. So this is one of the parameters you can think of. Even the sample processing method and lysing reagents. So different lysing reagents can affect the cell preparation also. So there are different RBC lyse. So you are using, suppose you are working on the bulk-like stain also, you should not use the lysing with fixative. So fax lyse also fixative, optilyse has a fixative, optilyse B and C both have the fixative. So you should go for the RBC lysing, that is without fixative, if you are working on the lyse wash and stain method. If you are stain lyse wash method, obviously you can use anything like fax lyse you can do. So we did one more experiment. These are the basically parameters. So cell to antibody cocktail ratio. So if you are doing on this working on the stain lyse wash, it's difficult to maintain, because to get the 5 million cells, you end up with something 1 ml sample or 800, depending on the count. It might happen you can end up with the 50 microliter of cell to get the 5 millions of cells. Even you can have the 100 microliter sample, 200 microliter. That can be wash it and you can make the palette and you can get the volume. Because if your sample to cocktail ratio is not maintained, again you are going to compromise with the antibody expression. Because your protocol is fixed on the 50 microliter sample, 15 microliter, everything is volume by volume. So decanting supernatant Christian PBS is difficult for the stainless wash. Bulk lyse is very good. No issue. Reagent validation like stainless wash do not have, because you are only fax lyse. You take a sample, you add the antibody, and you put the RBC lysing. Only parameter RBC lysing, and again you have to give the wash to get rid of RBCs. What are the RBC? So achievement of target size number is maybe, I don't know. Those who are working on stain lyse wash maybe up to 2 million, 5 million. I don't know. It's difficult, but for bulk lyse wash, you are confident even you are having okay. This I am like putting 6 million, 10 million cells. I am going to have the viables of 5 million cells. So additional fixative post staining is required for the bulk lyse stain lyse wash, and for the stain lyse wash already fax lyse can be used as a fixative. You can add for the TMRD, you can there are many more fix and pulse reagents from intraprep, perfixency, then BD-fix and perm, then from thermofisher, medium A and B. So that is again on your own experience, depend on the own experience. So what we run some panel along with all these fax slides. So we are for the first 4 plots for the stainless wash method. Last one is the bulk lyse stain wash. So we have done something like where finally we conclude in the summary that we'll just go through that. So you can only see on the fact slide, you can see there is a compromise on the fault scatter. All the rest are there is not compromise on the only being fact slides, there is a compromise on the fault scatter. Even in the scenario of fault scatter and side scatter, you can see only the fact slide has shown the compromise on the, so your cells are like, so you need additional adjustment for fault scatter to play with the gain to get you the proper data, FAC and SSC separation. So again, we have studied the 34% on the all the these factorialized, formalized, optilized, TMH like that is in our thing. This is a using the stainless wash. All these 4 are the stainless wash method. Last one is the bulk-wise wash method. So on the both these TMH light was used. So there is separation of B cell, T cells, and using the same cocktail, same sample, or for all the 5 different lysing reagents. So basically, this was acquired on the before any adjustment of the fault scattered, and this was acquired using the there is a for fax lines we did some gain experiment to get the proper FAC. That's why you can see all the forward scatter parameter on the first plot is matching with the agreement with the register. So this is again the same thing we did, and finally we come to the conclude that. So without adjustment of forward scatter and side scatter, we are losing on the percentage of 34. Like there is a either gain or loss of. So you can see there is always difference variation in the percent of B cell and cell cell if you are working on the fact slides basically. Except all and even fact slides on with addition of forward scatter and side scatter, B cell, the T cells are on the lower side compared to the all the things. This is our data. We are not like proposing anything. So you have to do at your in your own setup in your lab before you go for anything. That's our message. Even on the paper, you can see on the microscopically the farm lies, how the farm lies and the facts lies. That's why the reason is you get the false cutter and side cutter is compromised always. There is one paper on the CD45 and side cutter plot using all these immunoprep, optilite, versalite, the ammonium chloride base, EG-lite, UT-lite. So they have shown that there is a difference between the CD45 gated population if you use the different lysing method. One more important thing is concentration to volume of the antibody is very important. So optimal concentration antibody should be chosen. So for leukemia you might end up with some of the titer, but you might need additional volume of that particular antibody to deal with the 5 million, 10 million cells. Because whatever the number of cells compared to the ratio, that antibody may not be enough to stay in all the cells. So you ultimately you are ending with the expression of particular antibody. So which minimize the signal to noise ratio, minimize the background. So this for when you go for the MRD, that should be the volume should be the robust. Do not compromise any title to go for the cost per test, load on the cost per test. So this is how if you have end up with the high number of cells, so you might get both in both the scenarios you can see the 30th has been got because we always get 30th on the brighter side. This is how you can make it okay. The number of cells are very high. Sometimes instead of 5 million, the 10 million cells. So that is antibody is not always compromising only CD45, CD38. So when you use the polyclonal, this is one of the examples when you use the for myeloma or something. So polyclonal kappa lambda may not be used, but you have to go with the quad. So same case. So the previously that kappa lambda is the polyclonal kappa lambda, and we added after that the polyclonal kappa lambda from the goat polyclonal. So whether it is the kappa restriction has been said. So one more thing we can do to proper achieve data because in our MRD sample we have maximum part of the all the cells, including all the viable cells. The majority are the neutrophil. So if you can get rid of neutrophil using the phycol separation, so instead of having 5 million cells, if you have in this sample like the 49% of neutrophil. So if you can get rid of neutrophils, so you can end up with a 2.5 million cells instead of 5 million cells. So that can be also done, but that has to be again verified in your lab, and then you can go far. So along with the phycol separation, we might come up with the new things like you can do the bead sorting, like CD15 bead sorting. You can have the CD56 bead. So you sorting like basically you put the sample. You remove the neutrophils based on the 15 or CD16, then you process for your BMRD. So that can be done because majority of the time you get the non-specific binding because of these neutrophils, because of the many of the mesenchymal cells. These are the part of the MRD samples. So that's how you have to work to get the proper appropriate data. So these are the factors which are going to be affect your MRD, any types of MRD like DMRD, MLMRD, TMRD, what you're doing the MRD. So thank you so much.